Thank you for the introduction, and thank you for having me here, of course. I have basically divided my talk into three different topics. First of all, I would like to talk about methods for ChIP-seq enrichment data analysis. Then I will introduce into the actual study about enhancer mapping and primary human T cells. And finally, if time permits, I will very briefly mention what I'm doing today, what is namely chromatin mapping in pediatric brain cancers. So this is a very classical sketch about the ChIP-seq protocol. So we have our sonicated DNA fragments that have been enriched by an antibody specific for transcription factor, for histone modification, or any kinds of DNA modifications. Then we sequence these DNA fragments, map them to the reference genome, and get these pile-ups that tell us where in our sample of interest we do have our enrichments that we identify typically by using peak callers. But, of course, this is a very valid approach, but at least for me, in most of the cases, in most of the projects where I am involved, and we are not interested in looking at the epigenetic state of one genome, we are typically interested in comparing different conditions, like the wild type and the control or treatment condition here. And I would say a very naive approach to identify differential enrichment is to call peaks in the different conditions and then just say where we have peaks in one condition and not in the other would be our differential enrichment. This would work in an ideal world maybe, but most of the data clearly looks different. So we have these cases where we have an enrichment in the wild type, for example, and the peak caller didn't identify any enrichment in the control and in the knockout or in the treatment condition here. And the question is, of course, is there really no peak? So the thing is that transformation of ChIP-seq data into binary information loses very important information necessary for a quantitative comparison between conditions. Then there's a second artifact that can be seen here, that peaks can overlap in parts only. So there are the peaks span genomic regions with distinct epigenetic dynamics. So this is a very important observation that argues against another common approach where you first identify peaks in all conditions, merge the peaks and re-quantify afterwards. And finally, the peak finders typically do not consider variation across replicates. So therefore, this is actually what I propose to do. This is a quantitative comparison of ChIP-seq data with the aim of identifying statistically significant differentially enriched regions, which I call very broadly just DERs. They can be DMRs for differentially methylated regions or whatever. So differentially enriched regions. First of all, for each sample, calculate the sequencing coverage at genome-wide windows, perform quality control, saturation analysis, correlation between samples, and so on. Remove non- and lowly covered genomic regions, estimate size factors for library size normalization, calculate overdispersion, and for each window, then test the differential coverage between the groups across the replicates, correct for multiple testing, select significant windows, so that the DERs, and merge adjacent significant windows, and then there's a bunch of downstream semantic analysis. I have implemented this workflow into a bioconductor package that I have called MEDIPS. The name might be a little bit confusing. This is because I have initially developed this package specifically for assays sensitive for methylated DNA, so MEDIPS-seq experiments, and these assays have some known bias towards CpG density. So MEDIPS still maintains functionalities for correcting for CpG densities, but of course this approach of calculating differential enrichment is valid for any kind of DNA enrichment analysis. So we have generalized this package quite and improved the performance quite a lot. And it's now applicable, in my opinion, to many different kinds of DNA enrichment techniques. So now I would like to introduce a study where I have extensively applied MEDIPS. It is about epigenomic analysis of primary human T cells. So this is the experimental setup. We have 12 healthy subjects and we have 12 asthmatic subjects. So in total there are 24 subjects, and we have isolated from blood samples from these patients 3 different memory cells, T memory cells. On the one hand, the naive cells that never have seen an antigen, and on the other hand, TH1 and TH2, or CCR4 negative and CCR4 positive memory cells. In total, there are 72 different samples. Most of them have been analyzed in duplicates. And we were interested in mapping active enhancers and active promoters in these samples. So we have performed H3K4 Me2 enrichment in 120 samples. And of course, first of all, you're interested in how reproducible are your assays, especially because we have downsampled the number. We have tried to apply these chip sequencing assays to samples with very low cell numbers. In this case, we are looking at 10 million cells. So in general, here we are looking at a control region, at a housekeeping gene, where we do not expect any differences in the Me2 enrichment. And I'm showing you only the naive, or some of the naive and some of the TH2 tracks here, and they pretty much look like photocopies. Of course, we have calculated correlations and so on. So overall, the data is highly reproducible. And then I have applied the MEDIPS, the MEDIPS workflow. And again, I'm highlighting here the comparison between the naive cells and the TH2 cells, because the TH2 cells are actually those that have been, or that are known to be implicated in asthma disease. And in total, there are 50,000, around about 50,000 significant differentially enriched regions. And here we see the CCR4 locus, where we clearly see an enrichment in the TH2 cells. But I think it's also a good example that we see that the peak callers would have identified peaks in the naive and also in the TH1 cells. But when we look at the individual samples, we clearly see a significant enrichment throughout the TH2 cells samples. And of course, we're interested in how robust our data, how robust this approach is. So I have run I have randomly assigned the samples into random groups. I have applied MEDIPS many times, and in total, with the same adjusted p-value here, we see only 8 to 20 differentially enriched regions, leaving us with a very good false discovery rate here. So this is just a heat map of 71,000 differentially enriched regions when performing 3 cell type comparisons. So this is the set of distinct differentially enriched regions, what is approximately 1% of the human genome. And so in the columns, we see all the different DIRs. In the rows, we see the samples. And we have ordered the DIRs to highlight genomic regions that gain or lose either in both TH1 and TH2 cells, or specifically in TH1 or TH2 cells. Of course, we were interested in correlating these differential enrichment, this differential Me2 enrichment with gene expression. So I have identified based on matching RNA-seq data, differentially expressed genes. And we see those that are upregulated or those that are downregulated here. And then I have centered this heat map at the transcription start sites. And I think it is very obvious that we see concordant changes in gene expression and H3K4 Me2 enrichment patterns at promoters, but also at distal cis-regulatory elements. On the other hand, we also identify genes that have no significant change in gene expression, but they do change in promoter H3K4 Me2. So we believe that these could be genes that are actually poised for activation in response to further extrinsic stimuli. In an additional characterization analysis of our identified TH2 specific enhancers, we have observed that asthma GWAS SNPs are enriched, especially in TH2 enhancers. So what we have done is we have accessed the GWAS integrator database. We have downloaded all the available 82 lead SNPs. The problem with the GWAS analysis is, of course, that they are typically based on whole exome sequencing data. So all the non-coding regions, the intergenic regions, where we observe most of our differentially enriched regions, are not covered. So we have followed an approach by accessing haplorec, by identifying SNPs that are in tight genetic linkage. So we complemented our set of 82 lead SNPs by additional linked SNPs. And when we test how many SNPs overlap our DIRs, we observe that 52 potential asthma SNPs fall into 38 of our DIRs. And by a proper enrichment analysis, we observe that there is a clear enrichment of asthma SNPs, especially in our regions that gain Me2 during TH2 cell differentiation. So, of course, we were not only interested in the enhancer landscape, comparing the different cell types. So we were also interested in comparing our healthy subjects and the asthmatic patients to each other. So I have again applied MEDIBS to identify differentially enriched regions in 3 different cell types. We observe no DIRS in the naive cells. We observe no DIRS in the TH1 cells, but we observe a small number, so these 160 differentially enriched regions between healthy and asthmatic patients in the TH2 cells. And this plot on the bottom shows us where these DIRS are located, and shows some of the genes that are immediately affected by differential Me2 enrichment between asthmatic and healthy. So to conclude, to summarize, we have mapped H3K4 Me2 in naive CD4 plus cells and TH1 and TH2 cells from 24 different subjects. We have done a computational analysis of this large-scale ChIP-seq data, and we identified putative enhancer regions associated with TH1 and TH2 cell differentiation. We also observed that TH2 cell-specific enhancers are enriched for asthma-associated SNPs, and we identify enhancers associated with asthma susceptibility. Of course, I would like to acknowledge all the team, everybody who worked at this project. So these people are basically my former colleagues while I was working at the La Jolla Institute for Allergy and Immunology in La Jolla, San Diego. I have actually recently moved from La Jolla to the German Cancer Research Center in Heidelberg in Germany, where I am recently establishing a group on computational oncoepigenomics. So one major focus of my group is chromatin mapping in childhood brain cancers, and I think we have quite in our division for pediatric neuro-oncology, we have a huge data. We have many huge data sets available in different brain cancer entities. We are mapping different chromatin modifications. We complement this extensive ChIP-seq histone modification data with ChIP-seq data for selected transcription factors. We have always matching for the same patients, whole exome sequencing, whole genome sequencing, whole genome bisulfite sequencing, and RNA sequencing data available. And we are currently working in applying available but also developing novel technologies for an integrative analysis of all these data types with the aim to understand the relationship between the genome and the epigenome and how they collectively contribute to pathogenesis. And with this, I thank you for your attention.
Thank you very much for an interesting presentation and staying in time. We have time for 1 or 2 questions. Please.
Hello. I have 2 questions. First one is, in your method, since most of the current ChIP-seq data are run without replicates, so is your method able to handle those data without replicates to do differential enrichment?
Well, you can, basically, yes, you can run MEDIBS without having any replicates. Then the dispersion will be set to 0, and you will get probably overestimate of differentially enriched regions. So it's practically okay, but don't do it. It's better to have replicates.
Okay, and the other question is, have you compared your method with other published methods in terms of doing differential ChIP-seq?
Yes. So we have done some comparisons to other tools that came up at some time. And so, for example, here's diffreps and comparison to diffreps. I think we did another comparison to another tool, which I don't remember. So the bottom line is that, of course, the overlap is highly significant, so we are observing very similar DIRS, but on the other hand, it is difficult without having knowledge about the truth to decide which tool is really working better or which tool is really working was worse. So that was a situation where I was really happy to work with experts in the field of immunology, because they extensively looked for many days or weeks at the different results, looked at genes that they knew are important for memory cell differentiation. And the bottom line is what they told me is that, that there are many more true positives identified by MEDIPS, and that diffreps identifies many more false positives. This is all I can say about that.
Hi, I've used your tool a couple of times already and published papers, so thank you. I think it's a good tool.
Thank you.
The other question is that's popped in my head when using this. You know, you get the p-values on the individual window basis, and then you merge those. So it seems when you get these long stretches of highly significant regions, you're kind of understating the significance, I would think, in those regions. It's very, to me, compelling to see 50 or in sometimes really long stretches, maybe 50 is an exaggeration, 10 or 15 windows, each one with a very significant p-value, even after multiple correction. Have you thought about re-evaluating and merging those first and then estimating a combined significance rather than, because you're paying a big price for 6.6 million windows?
Yeah, that's true. So I don't know. There's one point. So this is a plot of the size distribution of the DIRs. So for example, we have many DIRs. The majority of DIRs are small windows, 500 or 1 KB windows or whatever. But there are, of course, there are some enhancer regions. Some people call them super enhancers, for example, that are longer stretches of differential ME2. So what MEDIPS has also a functionality not to divide the whole genome into small windows. This is one approach, but you can also do a focus analysis when you're, for example, interested only in gene regions, promoter regions, CPG islands, or whatever. You can simply upload these annotations and test specifically for these regions, or divide them into smaller bins and test for those. So what I recommend to do is then to start with the whole approach, merge adjacent windows, maybe even allowing for a gap size in between, and then using these merged genomic coordinates as an annotation to rerun MEDIPS and see if we can reorder the DIRs, the longer DIRs, then according to a new calculated p-values. This would be one approach.
Yeah, and one last comment. You've also pointed me to a package, an extension of this, the DIPSeq. I should call, what was it? Maybe it wasn't that name, but it was a colleague of yours who was developing it, and it was nice because you could also include covariates and paired analyses for before and after, so I thought that's worth mentioning because it allows it to extend to a general set of situations that others might adjust for age, if you're looking at methylation and whatnot. So.
That's absolutely true, yes. We are working on this.
Thank you.
Thank you.
Well then, let's thank Lukas and all the speakers of this session again.